Welcome to another episode from the Veterinary Research Channel. Today, we delve into a fascinating study published in the Journal of Veterinary Internal Medicine in 2024. This research, conducted by a team of veterinary scientists primarily from Chungbuk National University in South Korea, investigated the role of the complement system in dogs suffering from idiopathic epilepsy. Idiopathic epilepsy, often abbreviated as IE in scientific literature, but which we will refer to by its full name, is the most common type of epilepsy observed in our canine companions. To understand the significance of this research, let us first consider what idiopathic epilepsy entails. Clinically, it is defined by the occurrence of 2 or more unprovoked epileptic seizures that are separated by at least 24 hours. Importantly, these seizures arise without any identifiable underlying structural brain lesions or other signs of neurological disease. It's a condition that can significantly impact the quality of life for affected dogs and their owners. The prevalence of idiopathic epilepsy in the general dog population is estimated to range between 0.5% and 5%. Sadly, a considerable proportion, around 25 to 30%, of all epilepsy cases in dogs prove to be resistant to drug treatment. This challenge in managing drug-resistant epilepsy in humans has spurred investigations into various pathogenic mechanisms. One area of growing interest in understanding epilepsy is the role of the immune system. Examination of human brain tissue removed during surgery and studies using experimental animal models have highlighted the involvement of the immune system in the development and progression of epilepsy. A key aspect of this is neuroinflammation, which represents the specific interaction between damaged brain tissue and the body's innate immune system. Neuroinflammation can be triggered by classical factors like infections, toxins, and autoimmune reactions, as well as by events that increase neuronal activity, such as epileptic seizures themselves. However, if this inflammatory response is not properly controlled or spreads beyond the initial site, it can become harmful and contribute to the underlying mechanisms of the disease. Specific inflammatory molecules, including substances like interleukin-1 beta, tumor necrosis factor, cyclooxygenase, complement factors, and high-mobility group BOX1, are thought to significantly contribute to the development of epilepsy in otherwise healthy brains and to drug resistance in experimental models. Among the various pathways involved in neuroinflammation, the complement system stands out as a crucial effector of the innate immune system and also plays a role in modulating the adaptive immune response. The complement system is a complex network comprising approximately 30 plasma and cell surface proteins that interact with each other to initiate a cascade of inflammatory responses vital for defending against infection. Within this intricate system, the third component of complement, known as C3, and the fourth component of complement, known as C4, are 2 of the most abundant components that can be measured. The third component of complement occupies a central position where all the different pathways of complement activation converge, while the fourth component of complement is a key element of the classical complement activation pathway. Mounting evidence suggests that dysregulation of the complement system can contribute to the development of epilepsy. Notably, studies in humans have shown that the concentrations of classical pathway components are elevated in patients with epilepsy compared to healthy individuals, and are also higher in patients whose epilepsy is not well controlled compared to those whose seizures are managed effectively. These findings raise the possibility that these components could serve as potential diagnostic markers and targets for future therapies for epilepsy. The concept of a biomarker is essential here. It is defined as a laboratory measurement that reflects the presence or activity of a disease process. The discovery of a diagnostic biomarker specific to epilepsy, which could aid in diagnosis and classification, or a monitoring biomarker that could be easily measured in blood, would significantly improve the management of epilepsy in dogs. Currently, assessing the severity of seizures in dogs with idiopathic epilepsy relies on analyzing past seizure events, including the total number of seizures, how frequently they occur, and the intervals between them. As of yet, there isn't a readily available biomarker to objectively assess the severity of this condition in dogs. Given the established link between the complement system and epilepsy in humans and experimental models, the researchers in this study hypothesized that the complement system might also be involved in the neuroinflammatory processes in dogs with idiopathic epilepsy. Therefore, the main objectives of this research were to determine if complement dysregulation occurs in dogs with idiopathic epilepsy, and to investigate whether measuring the concentrations of complement components in the blood serum could serve as a diagnostic or monitoring biomarker for this condition in dogs. To address these objectives, the researchers conducted a case control study involving a group of 49 dogs diagnosed with idiopathic epilepsy and a control group of 29 healthy dogs. The dogs with idiopathic epilepsy were further divided into 2 subgroups: those receiving treatment for their epilepsy and those who were not currently receiving any treatment. For this study, The researchers measured the serum concentrations of the third component of complement and the fourth component of complement using a specific laboratory test designed for dogs, known as an enzyme-linked immunosorbent assay, or ELISA. Now, let us turn our attention to the key findings of this study. The analysis of the blood samples revealed a significant difference in the concentrations of both the third component of complement and the fourth component of complement between the dogs with idiopathic epilepsy as a whole and the healthy control dogs. Specifically, the serum concentrations of both the third component of complement and the fourth component of complement were significantly higher in the dogs with idiopathic epilepsy compared to the healthy dogs. When the researchers compared the subgroups of dogs with idiopathic epilepsy, those receiving treatment and those not receiving treatment with the healthy control group, they found that the serum concentrations of the third component of complement were statistically significantly increased in both the treated and untreated idiopathic epilepsy groups compared to the healthy controls. However, there was no statistically significant difference in the serum concentrations of the third component of complement between the treated and untreated idiopathic epilepsy groups themselves. Regarding the fourth component of complement, the overall group of dogs with idiopathic epilepsy had statistically significantly higher serum concentrations compared to the healthy control dogs. However, when the treated and untreated idiopathic epilepsy subgroups were compared to the healthy controls individually, the statistical significance was less clear. Notably, there were no statistically significant differences observed in the serum concentrations of the fourth component of complement between the healthy controls, the untreated idiopathic epilepsy group, and the treated idiopathic epilepsy group when analyzed separately. The researchers also investigated whether the serum concentrations of the third and fourth components of complement were related to various aspects of the epilepsy in the treated dogs, such as the duration of treatment and how well the seizures were controlled. For the purpose of this study, well-controlled epilepsy was defined as experiencing fewer than one seizure per month, while poorly controlled epilepsy was defined as having one or more seizures per month. Despite the fact that the time since the last seizure was significantly longer in the treated group compared to the untreated group, no significant differences were found in the serum concentrations of either the third or the fourth component of complement based on whether the epilepsy was well controlled or poorly controlled. In the group of dogs with idiopathic epilepsy that were not receiving treatment, the researchers explored potential associations between the serum complement concentrations and factors such as the duration of epilepsy, the time since the last seizure, and the frequency of seizures. Interestingly, they found that dogs experiencing seizures more frequently, specifically more than 3 times per month, had significantly higher serum concentrations of both the third and the fourth components of complement compared to those dogs with a seizure frequency of 3 times per month or less. However, no statistically significant correlation was identified between the concentrations of either the third or the fourth component of complement and the time since the last seizure in the untreated group. To further evaluate the potential of these complement components as diagnostic tools, the researchers performed receiver operating characteristic curve analysis. This statistical method helps to assess how well a diagnostic test can distinguish between two groups, in this case, dogs with idiopathic epilepsy and healthy dogs. The area under the curve provides a measure of the test's accuracy, with higher values indicating better performance. The results of this analysis showed that the serum concentrations of the third component of complement demonstrated good diagnostic accuracy in differentiating dogs with idiopathic epilepsy from healthy controls, with an area under the curve of 0.81. A similar level of diagnostic accuracy was observed when comparing the untreated idiopathic epilepsy group specifically with the healthy controls, with an area under the curve of 0.80. The optimal cutoff values for the third component of complement concentration were also determined, providing potential thresholds for distinguishing between the groups. In contrast, the serum concentrations of the fourth component of complement showed only sufficient diagnostic accuracy in distinguishing between dogs with idiopathic epilepsy and healthy controls, with an area under the curve of 0.60. No significant difference was found in the diagnostic accuracy of the fourth component of complement when comparing the untreated idiopathic epilepsy group with the healthy controls. Now, let us delve into the implications of these findings as discussed by the researchers. The study's most significant finding is that serum concentrations of both the third and fourth components of the complement system were significantly higher in dogs with idiopathic epilepsy compared to healthy control dogs. Furthermore, the observation that dogs with a higher seizure frequency had even greater elevations in these complement components suggests a potential link between complement activation and the ongoing seizure activity in these animals. These results support the idea that the complement system may play a role in the development or progression of idiopathic epilepsy in dogs, possibly through its involvement in neuroinflammation. While the exact mechanisms underlying idiopathic epilepsy remain to be fully understood, neuroinflammation has emerged as a significant factor associated with its development. Although prior research has indicated the presence of neuroinflammation in dogs with idiopathic epilepsy, This study is among the first to specifically evaluate the involvement of the complement cascade in this process. In humans, components of the complement cascade are known to contribute to inflammation within the nervous system by various mechanisms, including increasing the permeability of blood vessels, attracting immune cells, activating glial cells, and promoting the production of pro-inflammatory cytokines. Given that the expression of classical complement pathway proteins has been shown to be increased in brain regions experiencing neuronal loss and in reactive astrocytes and microglia, which are types of brain cells involved in immune responses, the activation of the classical complement pathway could indeed contribute to the initiation or maintenance of neuroinflammation in dogs with idiopathic epilepsy. The findings of the study align with previous reports in human patients with temporal lobe epilepsy and idiopathic epilepsy, which also demonstrated higher complement concentrations compared to healthy individuals. Therefore, this research suggests that dysregulation of the classical complement pathway, coupled with other inflammatory processes, is likely present in dogs with idiopathic epilepsy. The observation that serum concentrations of the third component of complement showed good diagnostic accuracy in distinguishing dogs with idiopathic epilepsy from healthy controls, while the fourth component of complement had only sufficient accuracy, is noteworthy. The researchers suggest that this difference might be due to greater variability in the serum concentrations of the fourth component of complement between individual dogs. In humans, variations in the number of copies of the gene encoding the fourth component of complement are a major determinant of its concentration in plasma or serum. Similarly, in dogs, the gene for the fourth component of complement is highly polymorphic, meaning it exists in many different forms. Consequently, the serum concentration of the third component of complement might be a more reliable tool for potentially aiding in the diagnosis of idiopathic epilepsy in dogs, whereas the levels of the fourth component of complement should be interpreted with more caution. The researchers propose that if the complement pathway is specifically activated only in idiopathic epilepsy in dogs, it could potentially serve as a specific diagnostic biomarker and even represent a novel target for therapeutic interventions. The study also explored the relationship between serum complement concentrations and the response to anti-seizure medication treatment. While the treated group had a significantly longer time since their last seizure compared to the untreated group, no significant differences were observed in the serum concentrations of either the third or the fourth component of complement between these two groups. Furthermore, no significant differences in complement concentrations were found based on whether the epilepsy was well controlled or poorly controlled in the treated dogs. This contrasts with some findings in humans, where complement concentrations have been reported to decrease with successful epilepsy treatment, and certain anti-seizure medications have been associated with changes in serum complement levels. The researchers suggest that the discrepancy between their findings in dogs and those in humans might be attributed to the differences in the types of anti-seizure medications commonly used in the two species. In humans, commonly used drugs like valproate, carbamazepine, and phenytoin are known to have direct effects on the immune system, influencing both humoral and cellular immunity and modifying the production of cytokines. In contrast, phenobarbital and potassium bromide, the most common anti-seizure medications used in the treated dogs in this study, have not been as extensively studied for their effects on the immune system. This difference in the commonly prescribed medications could potentially explain the lack of observed changes in serum complement concentrations in the treated dogs in this study. When examining the untreated group of dogs with idiopathic epilepsy, the researchers investigated the influence of factors such as the duration of epilepsy, the time since the last seizure, and the frequency of seizures on serum complement concentrations. The only significant finding was related to seizure frequency, with dogs experiencing more frequent seizures having higher levels of both the third and fourth components of complement. This finding is somewhat different from observations in humans, where seizure type and frequency have not been consistently correlated with serum complement concentrations. The researchers acknowledge a limitation of their study being the heterogeneity of the epilepsy samples with respect to prior drug treatment, although they attempted to mitigate this by performing comparisons within the untreated group. Interestingly, previous research in experimental epilepsy models has shown that levels of an inactivated form of the third component of complement in the hippocampus, a brain region often involved in seizures, correlate with the frequency of spontaneous seizures. This supports the notion that serum concentrations of the third and fourth components of complement might indeed be associated with seizure frequency in dogs with idiopathic epilepsy. However, the study could not definitively determine whether the increased complement concentrations are a cause or a consequence of the more frequent seizures. While it is possible that recent seizures could lead to an increase in complement concentrations, the lack of correlation between complement levels and the time since the last seizure in this study suggests that the elevated levels are not simply a direct result of very recent seizure events. These findings hint at the possibility that dysregulation of the complement system might contribute to poorer seizure control in these dogs. The researchers also point out several limitations of their study. Firstly, they characterize it as a non-hypothesis-driven orientation study, which meant they could not perform a power calculation to determine the optimal sample size beforehand due to a lack of prior quantitative data on serum complement concentrations in dogs with idiopathic epilepsy. The relatively small number of dogs included in the study could have also contributed to false negative results. However, the positive findings, such as the elevated serum complement concentrations in dogs with idiopathic epilepsy, provide a foundation for future, larger studies with more statistical power. Secondly, the study only measured complement components in the serum and did not investigate their levels in the cerebrospinal fluid, which directly bathes the central nervous system. While this study focused on easily accessible blood biomarkers, it is known that the blood-brain barrier, which normally restricts the passage of substances from the blood into the brain, can be dysfunctional and leaky in dogs and humans with epilepsy. Furthermore, alterations in serum complement have been observed in rats with chronic epilepsy. This supports the potential relevance of serum measurements, but future research could benefit from examining cerebrospinal fluid as well. Thirdly, the study only focused on the third and fourth components of complement and did not investigate other components of this complex system. Numerous systemic factors can influence the serum concentrations of complement components and other inflammatory proteins, particularly when only a single measurement is taken. Future research could benefit from a more comprehensive analysis of the complement system and its interactions with other inflammatory pathways. In conclusion, this study provides valuable insights into the potential role of the complement system in dogs with idiopathic epilepsy. The finding of elevated serum concentrations of the third and fourth components of complement, particularly in dogs with higher seizure frequency, suggests that complement dysregulation is associated with this common neurological condition in dogs. While further research is needed to fully elucidate the precise mechanisms involved and to explore the potential of complement components as diagnostic or therapeutic targets, this study represents an important step forward in our understanding of the complex interplay between the immune system and epilepsy in our canine companions. Thank you for listening to the Veterinary Research Channel. A link for the referenced paper is in the video description. Remember to test your knowledge by answering the questions in the description, and as always, keep those minds inspired, hearts light, and tails wagging.